Since the advent of DNA sequencing, we have gained the ability to construct more accurate evolutionary trees and to more precisely estimate the divergence times of species. Today, we'll break down the key ideas and algorithms used to achieve this. To deeply understand a new topic, I don't just read papers, I create computer models and experiment with them. This allows me to see and truly grasp the most subtle patterns. These very experiments form the foundation of my videos. To better understand the concept of the molecular clock, we first need to recall an important principle that plays a key role in evolutionary biology. If every organism in a population possesses a unique mutation at the same location in the genome, sooner or later, one of these mutations will replace all the others. If all these mutations are neutral or functionally equivalent, the probability for any single mutation to eventually replace all others is equal to 1 divided by the population size. Here, for simplicity, we are considering a population of haploid organisms. Let's imagine an organism that leaves exactly one offspring. With a probability of 1 in 250, a mutation arises in its offspring. It's not difficult to calculate that after 4,000 generations, approximately 16 mutations will occur. However, it's not entirely obvious what will happen in a large population of similar organisms, where genomes are shuffled due to sexual reproduction. How many mutations, on average, will one organism from such a population have? We will now try to model all of this. We'll take a population of 40 organisms and track a neutral segment 90 nucleotides long, where changes have no effect. Every newly emerged organism will receive a mix of the genome from 2 random parents from the previous generation. With a probability of 1 in 250, a new mutation will arise in a random location in the new organism. Here we go. 11 generations have passed. The generation count can be seen in the bottom left corner. 2 of our organisms have acquired a mutation. I mark newly arisen mutations with green. Repeated mutations at the same location will be marked with blue. In the next generation, these mutations will either disappear or begin to spread across that genomic position in the population. Since the population consists of 40 organisms and our mutations are neutral, only 1 in 40 of the newly arising mutations will be able to become fixed in the population. And here we see the first such mutation, which became fixed somewhere around the 300th generation. So, we are now dynamically observing the selected segment of the genome across the entire population and can see how new mutations become fixed in the gene pool. I actually ran 2 parallel simulations that started with the same ancestral genome. Think of them as 2 branches that originated from a common ancestor. In the meantime, let me describe the formula derived from the neutral theory of evolution. Let m be the number of mutations that arise in a newly formed organism. In this simulation, this is 1 in 250. Therefore, in a population of size n, the total number of new mutations arising will be n multiplied by m. Each of these mutations will become fixed with a probability of 1 over n. A mutation is fixed when it has spread throughout the entire population. We denote p as the number of mutations that become fixed in one generation. The population size in this formula cancels out, and we find that exactly the same number of mutations will become fixed in each generation as arise in a single organism, and the population size does not affect this. For diploid organisms, which have a double set of the genome, we would need to add a factor of 2 to the formula, but it would also cancel out. We have arrived at the final formula. The rate of neutral mutation fixation is equal to the rate of mutation occurrence per individual. This is the key conclusion: the speed of the molecular clock is independent of population size. For 1,000 generations have passed, so let's look at the results. In the left population, mutations became fixed 14 times, and in the right, 19 times. On average, this comes out to 16.5, which is virtually what was predicted. In the right population, 2 mutations occurred at the same site. The second mutation effectively erased the information about the first, which is why we only observe 18 mutations. The situation in the left population is slightly more interesting. 2 mutations occurred on top of older ones, but in doing so, they also reverted the nucleotides to their original ancestral states. As a result, only 10 differences from the ancestral variant will be visible. However, researchers do not have the ancestral genome, they can only compare the genomes from the left and right populations. It seems that the difference should equal 28. 10 mutations plus 18 equals 28. But in reality, 2 mutations in each population occurred at the exact same nucleotide position. Ultimately, researchers will observe only 26 differences in the genomes between the right and left populations. This is called saturation. The more mutations that have occurred, the higher the probability that a new mutation will occur on top of an old one, and there is always a chance that it will revert the nucleotide back to its original state. The only information researchers have is that in this segment, they can observe 26 mismatches between the species, and that mutations occur at a rate of 1 in 250 generations. The observed 26 differences must be corrected due to saturation. This is done using a special formula, which I will show you a little later. After correction, the number becomes 32. If these sequences differ in 32 places, we can assume that each species accumulated 16 mutations after diverging from the common ancestor. Consequently, the split into 2 separate branches occurred 4,000 generations ago. By knowing the rate of generation turnover, we can calculate the time when this event took place. This, essentially, is the core idea of the molecular clock. Now let's conduct one more experiment to confirm that population size does not affect the rate of neutral mutation fixation. I took a larger neutral segment and simulated enough generations for 50 mutations to theoretically become fixed. I ran 10 experiments each with populations of 40 and 2,000 organisms. This means the population sizes differ by a factor of 50. On the timeline, I marked the times when fixations occurred. As you can see, the number of fixed mutations is independent of population size. Since all of this is probabilistic, there is a small scatter in the number around the theoretical value of 50 mutations. Moreover, mutations don't appear uniformly over time, and if you take a small time window, the number of mutations can vary many times over. Therefore, for the molecular clock to work more precisely, it's advisable to compare longer genome segments, so that the number of fixed mutations is higher. This will reduce the scatter and bring the result closer to the theoretical prediction. And now, how the correction is performed. Let's assume one population split into 2 independent, sister populations, which then evolve independently. We will track a genome segment of 1,000 nucleotides. The black graph will display how many differences have accumulated in one population compared to the common ancestral sequence. The red graph shows the accumulated differences between the 2 populations. Ideally, these graphs should be linear and look like this. We start the simulation. At the bottom, I also show a graph of how genetic diversity, or polymorphism, accumulates within one of the populations. Light blue shows the maximum difference, while green shows the average arithmetic difference within the population. As you can see, the genetic diversity of the population remains at roughly the same level. This is a separate topic for experiments. Meanwhile, the graphs reflecting the differences between the populations deviate from a linear trajectory. This is the saturation effect of mutational substitutions. The fact is that mutations increase the genetic distance between populations. But the more substitutions that have already accumulated, the higher the probability that a new mutation will occur at the same place, on top of an old one. Moreover, a new mutation can revert the nucleotide to its original, ancestral state. Because of this, we underestimate the true number of mutational events, and our molecular clock begins to run slow. To correct this, we can use the Jukes-Cantor method. We plug the observed fraction of mismatches between the populations into the formula and calculate the corrected number of mutational substitutions. Using this formula, the results fall into place, demonstrating a linear dependence on time. I should note that in practice, researchers use more complex formulas which account for the different proportions of nucleotides in the sequence and the probabilities of transitions between various nucleotides. Note that the numbers 3 and 4 appear in the formula. Our uncorrected graphs will approach the value of 3/4 and remain at that level. This is because we only have 4 types of nucleotides. If you were to take random, unrelated sequences, on average they would match 25% of the time, and consequently, differ by 75%. We've now covered the idea of the molecular clock. Let's move on to the practical steps, the quality of which directly determines the accuracy of molecular dating. For comparison, we must identify homologous segments in different species, those inherited from their common ancestor. This is not an easy task. Not only have point mutations accumulated in these segments across different branches, but there also may have been insertions, deletions, translocations, and inversions, all of which complicate their identification. Once the sequences are chosen, they must be aligned. There are many algorithms that allow for the optimal arrangement of sequences underneath each other. I tried to implement one of the basic algorithms that allows for a global alignment of 2 sequences. Before the algorithm runs, scores are assigned for matches and penalties are set for mismatches and gaps. Then, according to specific rules, a scoring matrix is filled, and using a traceback path through it, we obtain the optimal alignment option, the one with the highest score. There might be several such optimal options. I should note that in practice, multiple sequence alignment is used, where sequences from many species are aligned simultaneously. Researchers often assemble sequences from various locations in the genome because the more material is analyzed, the more accurate the result will be. One approach is to use genes that are evolutionarily conserved and have been retained across many ancestors, and then use only the 3rd nucleotide of the codon for comparison. In the DNA code, every amino acid is encoded by 3 nucleotides. The substitution of the 3rd nucleotide is often synonymous, meaning it does not change the amino acid being encoded, and, consequently, such a substitution is neutral. A comparison sequence can be assembled from these individual nucleotides. This approach ensures the homology of the segments being compared. On a phylogenetic tree, we can date some of the nodes using information from paleontological excavations or known geological events that separated populations. By calibrating the clock against one or more nodes, we obtain the mutation rate for that branch and can then date the remaining nodes for which there is no fossil or geological data. In the video, for simplicity, I use the number of mutations per generation. In practice, the primary unit of measurement is the number of substitutions per nucleotide per year. This allows us to easily translate genetic distance into time in years, analyze branches with different generation turnover rates, and correctly compare research results, even if segments of different lengths were used for the analysis. And now we'll try to construct phylogenetic trees. There are several approaches to building phylogenetic trees. The most accurate and computationally expensive are the probabilistic methods, which are most frequently used in practice. But today, I will briefly describe 2 simpler methods, based on genetic distance and character comparison. To explain this method, let's run a small simulation. We have a population, and we will build a pedigree using a small segment of DNA that does not recombine and is passed on to the next generation along a single parental line. This will allow us to construct a lineage tree. Currently, the studied DNA segment is identical in all organisms, but one mutation occurs when passing to the next generation. This is our assumption for simplicity. Thus, after 20 generations, we should expect all representatives of the latest generation to have 20 differences in this genome segment from the original ancestral genotype. Now we will look at one lineage and try to reconstruct the entire history based on the number of differences, which is the genetic distance. We will count how many mismatches have accumulated in the genome between all representatives of the latest generation and record the result in a table. This gives us a genetic distance matrix. There are 2 mismatches between the 3rd and 4th organisms. This means each of them accumulated 1 mutation moving away from the common ancestor. Consequently, their divergence occurred 1 generation ago. We mark this on the tree. We do the same for all other similar pairs. Now, we treat each combined group as a single object. When calculating the distance from the new node to other nodes, we use the arithmetic mean of the distances to all members of the combined group. This is the averaging part of the method's name. There are 4 mismatches between the 9th organism and the group consisting of the 10th and 11th, meaning they diverged 2 generations ago. 2 other groups also diverged 2 generations ago. The common ancestor of the 1st and 2nd organisms lived 3 generations ago. There are 2 groups whose common ancestor lived 5 generations ago. 2 groups had a common ancestor 13 generations ago. Note that in one spot, we have 25 mismatches. A new mutation occurred on top of an old one. In reality, we would certainly apply a correction to this distance matrix before starting the process. And we are left with 2 independent branches whose common ancestor lived 16 generations ago. Knowing only the number of genome mismatches in the latest generation and the rate of mutation accumulation, we were able to construct a pedigree, indicating the exact number of generations ago when the branches diverged. However, we ended up with a lineage tree for individuals within a population. A phylogenetic tree for species is built using the same principle. Let's assume that after 15 differences in the genome, the lineages become reproductively isolated and can be considered separate species. We ended up with 3 different species, A, B, and C. Species A, separated from the others 16 generations ago. Species B and C diverged 13 generations ago. Here, we see the most recent common ancestor for each species based on the segment we are comparing. The greatest genetic diversity is observed in species B. This method is based exclusively on genetic distance. The method is very simple, fast to calculate, and immediately creates a rooted tree. Its main drawback is that it relies on the hypothesis of a strict molecular clock. The rate of mutation accumulation must be uniform across all branches. However, the rate can vary and depends on environmental conditions, the rate of generation turnover, and the efficiency of DNA repair mechanisms. Now I will describe the maximum parsimony method. This method, unlike distance methods, does not focus on the genetic distance, the total number of differences, but on the specific nucleotide substitutions at each site. Let's assume we have obtained the genomes of 3 closely related species and observe certain mismatches. We now need to construct a phylogenetic tree. Most methods build an unrooted tree, and for 3 branches, it will look like this. The root can be located at any of the 3 possible positions. To obtain a rooted tree, we need to add an external related species, known as an outgroup, which is definitely known to have diverged earlier. We will mark with color the sites in the genome where we see differences from the outgroup. We will call these characters. We now have a type of matrix. We can build 3 possible tree topologies. For each of the 3 possible trees, we will try to reconstruct the entire history of nucleotide changes. The pink character is present in all of them, so it most likely appeared before branches A, B, and C diverged. We mark this on every tree topology. Now we need to determine how the green character emerged. On the second tree, this is straightforward. The simplest scenario is when the green character appeared before the divergence of branches B and C. We can now observe this character in both branch B and branch C. On the first and third trees, explaining the green character requires a minimum of 2 evolutionary events. Scenario 1, the green character appeared independently in each of the two lineages. Scenario 2, the green character appeared at the very beginning, and then the nucleotide in branch A reverted to its original state. Both scenarios are equivalent as they require two events. We will stick to one of these scenarios. Now we mark on the trees where the blue character could have appeared. We sum up the events for all characters. The second tree topology is the most parsimonious, most economical. It requires the smallest total number of mutations to explain all the observed differences. Therefore, in the maximum parsimony method, this is the tree we select. However, this method has a major drawback called long branch attraction. The method does not account for the saturation effect. On long branches, mutations will frequently lead to the same variants arising independently at the same sites. The maximum parsimony method mistakenly interprets these as shared ancestry characters, synapomorphies, and groups these branches together, thus building an incorrect tree. Nevertheless, the maximum parsimony method remains a fundamental and highly effective tool for comparing morphological characters, especially in paleontology, where DNA sequences are unavailable and researchers can only compare external features and skeletal characteristics. A character matrix is created, and the most parsimonious tree, the one requiring the smallest number of evolutionary events, is selected. The maximum likelihood method and Bayesian analysis are the most powerful and accurate tools in phylogenetics. Unlike the maximum parsimony method, they don't search for the most economical tree, but the most probable tree. They utilize an explicit statistical model of nucleotide substitution, which accounts for the actual frequencies of nucleotides in the sequence and, most importantly, the different probabilities of one nucleotide replacing another. This allows researchers to obtain a much more reliable result. This is what the result of these methods looks like in a real study based on the exact same sequences. It is worth mentioning the selection of genome segments for comparison. For the molecular clock and for distance methods of phylogenetic tree construction, only neutral segments are suitable. It is in these segments that nucleotide substitutions accumulate at a roughly constant rate. For other methods, there is no such limitation, and the choice depends on the researcher. If species diverged recently, it is better to use neutral segments, as not enough mutations have yet accumulated in conserved genome segments to provide the necessary resolution. If species diverged a long time ago, saturation has occurred in the neutral segments, and it is preferable to compare conserved segments. Since finding the ideal segment for every case is difficult, in practice, researchers often combine both neutral and conserved segments to obtain the maximum amount of information about all time periods. We can construct similar evolutionary trees not just for species, but also for cells within a single multicellular organism. We can genetically label individual cells in the early stages of embryo development, to track exactly which precursor cells form various tissues, organs, and cell types. Or consider cancer research. By sequencing the genomes of cancer cells and comparing their mutations, we can reconstruct the sequence in which these mutations appeared and trace how the tumor evolved inside the body. And we can go even deeper. Inside the cell itself, there are thousands of mitochondria, and they undergo their own intracellular evolution. The principles we have discussed today are universal. They allow us to read the history of life at any level, from species down to a single cell. Evolution never stops, and thanks to genetics, we can date it and see it on all scales. That's all for today. Please like, subscribe, and leave a comment. Special thanks to everyone who supports me on Patreon. Bye everyone.